Good morning, everyone. Goutham Narla, Professor of Medicine at the University of Michigan in the Departments of Internal Medicine and Human Genetics, Secretary-Treasurer of the American Society for Clinical Investigation. An absolute honor this morning to talk to two incredible physician scientists, Dr. Arul Chinnaiyan and Dr. Charles Sawyers, recipients of the 2026 ASCI Harrington Prize for Innovation in Medicine. This honor awards physician scientists who have moved science from the bench to the bedside and transformed the care for patients suffering from human disease. I'll start by introducing Dr. Sawyers. Dr. Sawyers is the Marie-Josée and Henry Kravis Chair of Human Oncology and Pathogenesis at the Memorial Sloan Kettering Cancer Center, an investigator of the Howard Hughes Medical Institute, a winner of the Lasker Prize in Medicine, whose fundamental discoveries have transformed the care of both patients with leukemia as well as prostate cancer. Dr. Chinnaiyan is a professor here at the University of Michigan, a member of the Howard Hughes Medical Institute, AACI member, AAP member, and director of the Translational Pathology Center here at the University of Michigan, who's made seminal discoveries in the identification of fusion proteins that drive solid tumors and the ability to target them therapeutically. Both world-renowned physician scientists and at a personal level, people who I've always looked up to as a physician scientist myself. They've really inspired many of us to pursue the pathways and careers that we've chosen. And so it's an honor this morning to be able to speak to them. Dr. Sawyers, Dr. Chinnaiyan, thank you.
Thanks for having us. We're really excited to have this chat.
Thanks a lot, John. Dr. Sawyers, if I could start with you, you've done something that very few of us, if any of us, can aspire to, which is you've made seminal discoveries that have transformed the care first in patients with chronic myelogenous leukemia, and then subsequently prostate cancer as well. Can you tell us a little bit about that journey and how you're able to pivot and be able to basically make discoveries in two different fields at that level?
Well, thanks for the question. I wish I could say that it was a planned, pivot, but things just happen. When I went to medical school, I didn't know I wanted to be a scientist initially, and I sort of got bit by the bug when I was assigned my roommates. As a first-year medical student, I was in a quad with a bunch of MD PhD students, one of whom both of you know, Bill Nelson, who's the cancer center director at Hopkins. And about halfway through the first year, I was like, what am I doing? I should probably be doing this. So I didn't switch into the MD PhD program, but I did just sort of become passionate about science. And that's the reason I'm telling you that is because I became fascinated with leukemia as not only a topic in medical school and the biology and the translocations and so forth. It just seemed like the disease where the chance to molecularly understand a driver alteration, as we call them now, was possible. And then I intentionally sought out Owen Whitty's laboratory at UCLA after he was the first group to clone the BCR-ABL translocation in chronic myeloid leukemia. And I kind of got off the clinical training track and became a postdoc in his lab for 5 years. And that's how I got interested in kinase signaling. And then Gleevec appeared, and it was an incredible time, as I think everyone knows. And to get back to how did I pivot to prostate cancer? Well, the work was in Gleevec was going really well, and one of the main problems that presented itself was the problem of acquired resistance. And I think, as probably some listeners know, we were able to sort of decipher that problem by sequencing patients who were responding and then during remission and relapse, and we've discovered kinase domain mutations as the cause of resistance. That led, I think, as some people know, to next generation drugs. And so the problem is not completely solved now, but patients with chronic myeloid leukemia have a normal lifespan at this point. And I was in my mid to late 40s, and I was like, I got to work on something else. So it's not exactly as clean a pivot as that, but I thought, what is another drug resistance problem that because I felt like I had kind of a way of thinking about it that wasn't really, I mean, it seems obvious now, but it wasn't so prevalent a way of thinking. So I started, pivoting to prostate cancer to ask the question about hormone resistance.
Thank you, Dr. Sawyers. Dr. Shine, to build upon that, Dr. Sawyers and others identified key drivers, fusion proteins that drive hematological malignancies, and you went on to identify some key ones in prostate cancer. Can you take us a little through that journey and what kind of allowed you to kind of think maybe a little of outside the box that the same paradigms that applied to hematological malignancies might also apply to certain solid tumors as well?
Yeah, Gautam. So I think my training path, I think, was somewhat unusual. As you know, I pretty much completed essentially all of my scientific and medical training at one institution, the University of Michigan. I started here as an undergrad and then pretty much never left. Michigan, I think, provided an extraordinary environment, I would say, for both clinical training and scientific exploration. And then I would say my research trajectory, I guess, really took off during my MD PhD program in the medical scientist training program. I did my graduate work in the laboratory of Vishva Dixit, who was then a professor of pathology at Michigan, before he moved on to Genentech Roche. I guess in Vishva's lab, I was fortunate to be part of an early effort to really define the components of programmed cell death or apoptosis. This was a very exciting time for me scientifically. I had the opportunity to clone and characterize a number of key components of the cell death pathway, including FAD and lyse, better known as caspase-8 nowadays, as well as the death receptors DR3 and DR4. That was, at least as a formative thing for me, was certainly a magical time in the research laboratory. I would say that after completing my PhD with Vishva, I returned to clinical training and ultimately chose pathology for my residency. And at the time, the chair of pathology was Peter Ward, who also served on my PhD committee, interestingly enough. He offered me an opportunity to start my own laboratory here at Michigan while completing my residency. So I thought that this was an extraordinary opportunity and one of the major reasons that I continue to really stay at Michigan. When I began my independent laboratory, I became very interested in emerging genomic technologies, bioinformatics approaches, and this was pre-next generation sequencing era. And this is when cDNA microarrays were all the rage, and it was really the most powerful tool at the time to really study cancer biology, at scale. So I was one of the first investigators to establish cDNA microarray technology here at the University of Michigan, techniques that I learned from Pat Brown's lab at Stanford. With those tools, my lab and many others began to profile human cancers to identify patterns of gene expression to reveal new biology. And around that time, my lab developed this resource called Oncomine, a cancer microarray database analysis platform. Oncomine really allowed us to systematically compare gene expression patterns across many tumor types and to look for genes that are highly abnormal or had an outlier expression in a subset of patients. And I think that led me to naturally, I guess, work on prostate cancer, primarily because Michigan had a strong prostate cancer program, I would say, including well-annotated tumor biorepositories. We had an NCI spore program, which made it really an ideal setting, I would say, to apply genomic technologies. We began to profile prostate cancer samples using that microarray technology. And by combining that data with this outlier analysis approach, we developed, through Oncomine, identified that ETS transcription factors showed this striking outlier expression in subsets of prostate cancers. And that observation really prompted us to ask why these ETS genes were so highly expressed in certain tumors. Scott Tomlins, who was a talented MD PhD student in my laboratory at the time, led up the follow-up work using 5' race technology to characterize the upstream sequences of these transcripts. And that work then sort of led to the, I would say, serendipitous discovery of recurrent TMPRSS2 ETS gene fusions in prostate cancer, and particularly TMPRSS2 ERG, which is found in a very large fraction of patients. And certainly at the time, it was a surprising and, as you indicated, a paradigm-shifting observation. Before then, recurrent gene fusions were largely thought of as defining lesions in hematologic malignancies, such as chronic myeloid leukemia, a disease so beautifully studied by Charles Sawyers and others. The idea that common epithelial tumors like prostate cancer could be driven by recurrent gene fusions was not really appreciated at the time. So I would just say in general, my path to studying these recurrent genetic defects in prostate cancer came from the convergence of a few things: training in molecular mechanisms of disease, clinical pathology, early adoption of genomic technologies, access to exceptional prostate cancer specimens at Michigan, and maybe a willingness to let unbiased data really point us toward unexpected biology, I would say.
Thank you, Dr. Nye. So for both of you, maybe start with Dr. Sawyers. I mean, one of the things that's been truly inspirational for all of us about your careers is your strong focus on basic discovery science, but with an eye towards translation. And perhaps you could speak, especially in this current environment, about the importance of just basic mechanistic science and its role in drug development. And perhaps the second part of that question for both of you is, how do we couple that discovery to true translation to our patients and the role of, for example, venture capital and industry in that process?
Well, I mean, obviously, it means a really important question because of today's climate in particular. There's no doubt that everything that I've done translationally was built on a foundation of basic discovery that in many cases, went back 20, 30 years. I think for young physician scientists who might be listening to this, it's important to not just read those papers and act on them. It's to get the experience of doing basic science yourself, because I think you really need to understand what's under the hood. You can't just read the abstracts and sort of I mean, I just go back to what the story Arul just told. I mean, so I was a heme malignancy guy because that's where the translocations were, and why were they there? We thought it was something about heme cells. It was because we could grow those cells in culture and see them and do metaphases. But to Arul's credit, that wasn't possible with prostate cancer or other cancers. But he used another way of thinking about it. Arul, the way you told the story, which is the way I understand it when I first met you, you didn't think it was a translocation. You just used,
That's right.
clever observation to go, gee whiz, why is this thing so upregulated? So I mean that maybe that's the best example. If the technologies to do that kind of gene expression array analysis hadn't been invented through a lot of basic, even engineering, technologies, we wouldn't perhaps know what we know today. So that's the part about basic science. Maybe I'll throw it to Arul for whatever you asked next.
Sure. I mean, Goutham, unless you wanted me to follow up on a different question, but maybe I could sort of follow up a bit in terms of a team project that I had an opportunity to co-lead with Dr. Sawyers in terms of the Stand Up to Cancer Prostate Cancer Foundation Dream Team, which really I think began with, I would say, sort of foundational basic science that actually then translated into some intriguing clinical observations that have had patient impact, but really wouldn't have been possible without some of the foundational work in terms of profiling metastatic castration-resistant prostate cancer. And certainly, I've been a long admirer of Charles's work. I mean, I think his contributions in really understanding CML, I mean, a disease driven by a recurrent gene fusion, I think were foundational in terms of understanding the resistance mechanisms. And his particular pivot into prostate cancer, which is also a gene fusion-driven cancer. And certainly, I think our work there's been a natural connection, I would say, between our work, primarily because the gene fusions we identified in prostate cancer, particularly TMPRSS2-ERG, are androgen regulated. So in a sense, androgen receptor, which of course testosterone, the male hormone utilizes, is directly linked to the expression of these recurrent gene fusions as important drivers of prostate cancer. And when you therapeutically impact androgen signaling, you're essentially shutting off the gene fusion. I would say that our efforts in terms of the precision oncology space, I think, converged with Charles' efforts, especially in this area, was really sort of initiated through our first clinical sequencing programs for advanced cancer that we called MyOncoSeq, that we developed here at the University of Michigan. And this is an effort to really, in the next generation after microarray technology, sort of bring on next generation sequencing and how that can be incorporated into precision medicine workflows, starting with tumor sequencing, integrating germline sequencing, and then returning those results to patients. So I think one of the exciting team projects that I've had the fortune to work with Charles on has been this Stand Up to Cancer Prostate Cancer Foundation Dream Team. And this allowed us to really take these precision medicine approaches really at scale across multiple institutions. I think the Dream Team itself was one of the first major multi-institutional precision oncology efforts in prostate cancer. It brought together leading groups across the US and the UK, linking clinical expertise, genomic sequencing, pathology, bioinformatics, translational research, I would say in a coordinated way. But one of the most important aspects of the project was it was focused on patients with metastatic castration-resistant prostate cancer. And at the time, this patient population was essentially, very little was known about this patient population because these samples were harder to get at than, for example, clinically localized prostate cancer. And what we found through this team project was that advanced prostate cancer wasn't a single molecular entity, but rather a disease with distinct genomic subsets that could have therapeutic implications. I would say one of the most impactful, though, findings from that effort was that a substantial fraction of the patients, on an order of about a quarter of the patients, had alterations in DNA repair pathways. This included genes such as BRCA2, BRCA1, mismatch repair genes, CDK12, and others. And interestingly enough, a good fraction of those alterations were actually occurring in the germline. So it had really implications not only to the patient, but the patient's family in general. But in terms of sort of a kind of foundational observation then being translated into clinical impact, I think this is where sort of this Stand Up to Cancer Prostate Cancer Foundation team effort really had the impact, because it really laid the foundation for PARP inhibitors in metastatic castration-resistant prostate cancer, in that patients with BRCA2, BRCA1, and related DNA repair genes could potentially benefit from PARP inhibitors. So I guess more broadly, the Dream Team really showed the power of a collaboration at scale, not only moving foundational observations to translational impact, but it really allowed us to sort of work together in a major collaborative effort.
Can I just add to that? Again, for maybe some of the younger listeners, there was no hypothesis behind that project. It was a technology, it was a moment in time when technology and then the opportunity to use the resources of the Stand Up to Cancer and Prostate Cancer Foundation, philanthropy, to do something that needed to be done. As Arul said, the Cancer Genome Atlas had sequenced, 500 localized prostate cancers. So we said, let's sequence 500 castration-resistant prostate cancers, but we really didn't know what we would find. So I think I guess my point in bringing up the point I'm trying to make is, in a case like that, sometimes big science is the right solution, and then small projects or big singles will come out. And the BRCA observation is unbelievably important clinically, and eventually we would have found it, but it's really great. Another thing in big science projects like that Arul and I, did as we built the kind of community of people, you have to get a buy-in into data sharing and credit and all that kind of stuff. So we spent a lot of time thinking about that. and giving younger, members of the team the leads on different papers and different sort of spin out stories and so forth, presentations at meetings. And so there's a whole sort of, I would say generation of what were like the assistant professors and senior postdocs from that work that are now leaders in the prostate cancer field.
Thank you. But maybe I could just expand on that. And given that as we trained, some of our science was perhaps more limited by the technologies that we had. And Arul, Dr. Chinnaiyan alluded to race and other things at the time that were cutting edge, but now we can do next generation sequencing at scale and whole genome sequencing, and now with generative AI and such. How do you feel that like technology and for our trainees, and how do we integrate that into science while still staying true to our purpose and really understanding that fundamental mechanism-based science is still critical for translation while utilizing state-of-the-art tools that we have available to us today?
Well, I don't have a great answer for the AI part because I feel like I'm almost too old to learn that, but I do have a Claude account, etc. And I do serve as an advisor to a biotech company that has its goal as to use AI, drug design, and I have to say, it's not clear that undruggable targets suddenly are solvable with this technology. I mean, because as we all know, the power of this is pattern recognition that humans can't see, and so if there's no knowledge about a target that's undruggable, I don't see how it can just sort of be dreamed up, but that we'll see. But I guess the point I would make is you need to partner with people who have technological savvy to, science is more collaborative than it ever was by necessity because I don't think anyone can be great at all of these, different tools, but they all are critical to be brought to bear on the questions that we're interested in.
Yeah. I would just add that certainly I think AI and enhanced AI approaches are certainly enabling and allowing sort of others that are sort of playing at a certain level of expertise to sort of bring in diverse expertise by taking advantage of AI approaches. And that's certainly the approaches that we're exploring. But it's, again, sort of early days. And I would still say that it's exquisitely important to focus on understanding biology and mechanisms of disease. I think there's only so much that AI is going to really help in that context. But I sort of see it as sort of an enabling tool more than anything else.
And perhaps just lastly, I mean, as advice, you're both products of incredible mentors, Dr. Dixit, Dr. Owen, and many others. And maybe for the next generation of trainees who might be listening to this, what are the lessons learned perhaps, and what are the most important lessons you'd like to perhaps discuss or tell them to really do the incredible things the two of you have done and inspired so many of us to do science?
Well, I guess in my case, it was, going to a lab where, where it's just the lab is on the cutting edge of something exciting that you really want to be part of. And you've got to show up in a major way, which is never, that's for anything, I guess. And I think in my case, I learned a lot from Owen. I also learned a lot from the other people in the lab. So I think it's important to join a lab that has a critical mass. Giant labs, I think, are probably a mistake, but maybe not. But I think really small labs, I worry that it's hard to get enough experience because you learn just as much from your sort of failed experiments. And you learn how to get around those, not only from your mentor, but from the people around you at the bench. And if it's not in your lab, it's in the lab next door. And so talking to people all the time during your training about your work, as long as they'll listen to you, you have to learn your elevator pitch to get them excited, and also be really interested in their work, because it's incredible how much you learn about your own project from seeing how someone else solves a problem in their story that seems remotely, related, but may have a critical kernel of insight.
Yeah, I guess I could add to that just based on my experience. I think one lesson that I learned was that um, career paths don't have to really be linear, I guess, to be successful. I guess in my case, I essentially did all of my training at the University of Michigan, which is unusual. But what mattered was not really changing institutions for the sake of change. It was really finding new scientific environments, new mentors, new technologies, and new problems that pushed me to sort of keep growing, I would say. And as you alluded to, Gautam, I would say that mentorship was absolutely critical. I was fortunate to have extraordinary mentors at different stages of my career. I mentioned Vishva and Peter Ward, many others who I think gave me the scientific freedom, challenged me intellectually, and created opportunities for me. And I try to pass that forward to my own trainees, I would say. Um, I think through my experience, I think I also learned the value of being open to unexpected directions, especially in the context of the discovery around the prostate cancer fusions. I think many of the most important opportunities, I would say, in science come from following the data, even when it takes you somewhere that you really didn't expect. Then in terms of sort of combining MD and PhD training, I would say that my clinical training in pathology also shaped the way that I think in terms of seeing disease through the lens of patient samples, diagnosis, and clinical need. I would say that for our trainees, I think it'll be important to stay connected to human disease that you're studying, and whether you're a physician scientist, basic scientist, computational biologist, or a translational researcher. Also, I guess for the trainees would be advice around choosing an important problem. Again, techniques and approaches, AI approaches, all of this will change over as the field evolves. But if you're working on an important question, I think that matters to you deeply, the biology and translating that in terms of medicine, ultimately to patients, I think you'll have a reason to keep on going, I would say. And then perhaps lastly, I think being comfortable crossing boundaries, especially with the advent of AI approaches nowadays. I think some of the most exciting advances really happen at this interface of disciplines in terms of, for example, in my case, pathology and genomics, computation, clinical oncology, chemistry, and cancer biology. So I think trainees that can kind of speak in more than one scientific language, I think will be particularly well positioned in today's age.
Like to thank you both. I mean, your journeys are inspirational at a personal level, I'm sure at the level of our entire academic community, to make seminal discoveries and to translate them into meaningful drugs and applications for patients, our patients, is remarkable. Congratulations on receiving this milestone award. And thank you as well for your mentorship of so many and the inspiration that you've provided us.
Thank you.
Thank you so much.